Thank you, Tom. You may begin.
Thank you for the introduction. Good morning. Today I will be discussing my project that focus on a new way to quantify protein structure using laser-induced fluorescence. This is a collaborative project that involves Dr. Michael Cassio and Dr. Theodore Kirkovolos as the principal investigators. So the main goal of the research is to improve the methods used to study protein structure. The three standard methods that are commonly used are X-ray crystallography, NMR spectroscopy, and cryogenic electron microscopy. And while these methods are great, they do have some limitations, mainly in that most of them require them to be static and frozen, as well as requiring a large amount of sample. Cross-linking mass spectrometry is another method that's used in the Cassio lab that complements these high-resolution techniques and can be conducted in full-length proteins in the presence of a membrane. But the one drawback with this method is that the output isn't quantifiable. So what we wanted to do this summer was optimize a laser-induced fluorescence platform for the protein quantification. So first, I just want to take a moment and describe what is fluorescence. So essentially, fluorescence occurs when a molecule absorbs light at one wavelength and releases it at another. We can see on the diagram here, we have our incoming light coming to our molecule. They'll bring it to an excited state. And just through natural phenomena, the energy will be slightly less. And it will release a wavelength different than the absorption. And this is what's known as fluorescence. So in our system, there's about a difference of 65 nanometers. And then the actual quantification that we're looking for comes from the fact that we're going to have a photon counter that'll count the fluorescent photons. And so that the number of the emitted photons will be proportional to the concentration of the sample that we're looking at. So here's how exactly we're going to get our fluorescence. We're going to use a molecule known as naphthalene 2,3 dicarboxylic aldehyde, also known as NDA. It's kind of a mouthful. This is going to be a fluorescent tag. So what it's going to do is we're going to mainly look at the peptide sequences of the glycine receptor. So what NDA does, it will label the primary amines in the peptide sequence of the glycine receptor after a process known as trypsinalysis, which is an enzyme that will cleave the peptide bonds. So the NDA is going to be tagging the peptides at the N terminus and at either the lysine or an arginine. So what's interesting is that the NDA will only fluoresce when it is binded to our peptide sequence. So, and then the more product that's present, we'll have a stronger fluorescence. Okay, so another key feature of our platform is the idea of microfluidics. So a huge benefit of microfluidics is that it requires a very small amount of sample and thus will have a quicker reaction time. So reactions will occur in these droplets of carrier fluid, and it's going to be in the form of like an oil droplet so that it doesn't mix with our reactants. And it'll go through these tunnels. And as it goes through the tunnels, the reactants will stretch and fold, thus making the reaction occur quickly. So as you see here, it'll go through all these tunnels, like all these channels, and then right at the end, we'll take our measurement. So each droplet will only carry about 5.4 nanoliters of solution. So here's the diagram of our actual setup. We're going to have an ultraviolet laser that will get reflected onto our sample via a 40 times microscope ejective lens. And then it will then go through a dichroic filter to make sure that we only get our emitted light. And then we'll go into our photodiode detector. And then here's a picture of the diagram in the lab as it is today. So here we show our ultraviolet laser going to our sample, which is on our microfluidic chip. and then we have our fluorescent light that goes to our photodiode detector. So one of the main objectives this summer was to produce new microfluidic chips, and we did so using a chemical known as polydimethyl 6,6-siloxane, also known as PDMS. So the first image we have here is the silicone disc with a pattern of the chip. So using the process of photolithography, light was used to transfer the pattern from a photomask onto our silicone disc. We then poured PDMS onto the disc and removed any air bubbles that would have been present via a vacuum pump. And then we put that disc covered with PDMS in the oven, about 75 degrees Celsius overnight. And then we scraped off the PDMS covering, and then we cut off the individual chips and bonded it to a glass slide using a plasma chamber. So in summary, the main objective of our project is to use fluorescence as a quantification tool to examine sample concentration. The reactions occurred on the newly made microfluidic chips, and their main benefit is they require a small amount of reactants, so we can efficiently run our reactions. And then we would like to run this platform along with the cross-linking mass spectrometry to obtain both the quantification and the identification of glycine receptor samples. So in the future, we would like to calibrate the fluorescent detection as a function of composition and determine the limit of detection. We would also like to measure the reaction rate versus flow rate to see exactly how much sample we need to run a complete reaction. And then the overall goal is to integrate with the cross-linking mass spectrometry. To get more information on that side of things, like how the peptides were made and how the preliminary data, please see Thomas Durrat's talk at 11:50. His abstract number is 24 in the booklet. And these are all the acknowledgements that I would like to thank for either helping work on the project or for providing funding for the research. Thank you.